Next up we have the apparently multi-talented Vivek, who is sporting an attire that I can only assume either he's made himself or purchased on some niche corner of the internet.
No, I did it myself.
But I urge you to ignore all of that, at least temporarily, and instead focus on what he's saying about balancing.
Well, thank you. Hi, my name is Vivekanand. I'm part of the Ideas and Spatial Orientation lab. And today I've got two objectives to this talk. The first objective is a general objective that you do, is to give you an overview of the research that I'm doing. But recently, since I've become a postdoc, I've had a hunger, I've had a dream that I want to collaborate, I want to idea generate, I want to mix with people that I typically don't mix with. So I'm also going to focus on holistic ideas where you and I can potentially collaborate on. And some of you might be thinking, that's impossible. This guy works on human spatial orientation. It has no relevance to what I do in my research. And the hope is that today I'll show you some fertile grazing grounds where you and I can eat together and say, look, there's some really cool ideas that exist between us. So the actual work I work on is the role of gravitational cues in the learning of human balance control. And the way I study it is I strap people into this mechanical beast known as the Hulk. And the Hulk is programmed to behave like an inverted pendulum. An inverted pendulum is just like trying to balance a pen on your fingertip. If you do nothing, it just naturally falls to the left and right, which is what's happening here. There are also crash boundaries. So if you go too far, the machine stops and automatically resets the balance point and it starts again. We chose inverted pendulum dynamics because it's a good first approximation for human balance and postural balancing in general. Now what subjects do is they grasp this joystick and they try to stabilize themselves at the balance point. And what you'll notice is that because they're seated and because they're using a joystick to control the motion of the machine, they cannot use the peripheral mechanisms that we typically use to balance. So the biomechanical properties of your legs you cannot use. And you cannot use the reflexes that typically we use to balance. And so it becomes an interesting window into central processing of balancing. And I'm not going to talk about this perspective too much today, but if you're curious, ask me at the end and I'll tell you more. Now, balance control depends on many different sensory systems because balance is very complex. In order for us to have robust balancing, we have to have all of these sensory systems layered on top of each other. And when you remove one or two of these sensory systems, people encounter illusions that, for example, to military aircraft pilots can lead to fatal accidents. In my experiments, people are blindfolded, so they cannot use their visual system. They wear noise-canceling headphones, so they cannot use their auditory system. And because they're strapped into this machine, they really can't use their proprioceptive system to figure out where they are. They're relying primarily on their vestibular system, which I'll describe in a second. And their somatosensory system, which consists of touch receptors on their skin. As they push against that machine, they get some information about how it's moving. Now, the vestibular system, many people are not intimately aware of. It resides in your inner ear. And the vestibular system is comprised of two components. The first are your semicircular canals, which are filled with fluids. And what they detect is angular acceleration during rotation. And as a result, they are getting motion cues. Every time you rotate, you get a sense of your motion. The second component of the vestibular system are your otoliths. And your otoliths are these fine little hairs that are suspended in gelatinous material adorned with crystals. And what they detect, among other things, is tilt from gravitational vertical. The more you tilt from gravitational vertical, the more these hairs deflect, informing you that you're tilting. And so as a result, they provide positional cues. Now in my first condition, the upright condition, Where people are in the upright, the machine is falling to the left and right. If they tilt a little bit, as I just described, those otolith hairs tilt a little bit, informing them. If they tilt even more, let's say by 45 degrees, again, they're blindfolded, but they have a good sense of their position. As a result, from your otoliths, you're getting position information because you're tilting relative to gravity. From your canals, you're getting motion information. And from the somatosensory system, you're getting a mix of both of them as you're pushing against that chair. And the second condition, on the back, I tilt people backwards, and now the vestibular system, or the vestibular signal, is a little bit different. When they're on their back, the dynamics are all the same inverted pendulum dynamics. But how is the vestibular system slightly different here? Let me explain. When you're on your back, those otolith hairs are deflected back, saying, hey, you're on your back. And whether you're pointed here, or here, or here, the otolith information is always the same. You don't get any extra information about your angular position because you're always just 90 degrees from gravitational vertical. So this becomes a really unique way to look at a system where you no longer have any positional information. All that you have is motion information. And so the underlying question that I'm looking at here is, are motion cues sufficient for a dynamic stabilizing task? Another way of interpreting that is, can people even adapt to the absence of relevant positional information? And these words that are in bold and in red are words that link to Katya's presentation, because we met for lunch and I was like, I want to link my presentation to yours. And this is one of the words that she had used. Now, we can go into the literature to get some insight. And what the literature says is that the canals are taking angular acceleration information and they're integrating it into a velocity signal. This is pretty established. Not too many people debate it. Then the next idea is that that velocity information is sent to a neural integrator. And that neural integrator makes an estimate for position. Because if you have velocity and time, you can make an estimate of position. And there is experimental evidence that supports that this is entirely possible in the human being. And if you're curious about these set of experiments, you can ask me at the end. So what I'm going to do now is show you some quick data on the upright condition, where people know where they are, compared to the on the back, where they don't have a strong or any positional information. This is data for people in the upright condition. On the x-axis is block number, early in the experiment, and then this is late in the experiment. And again, in my experiments, people just balance for 20 trials, and they're just trying to balance. That's it. No real difference. I'm looking at learning. And on the y-axis is standard deviation for angular position, which is just a measure of variability, another key word that Katya had used. Early in learning, the variability is quite high. By the end of the trial, the variability is quite small. So when you have positional cues, people show learning. Nothing too surprising here. However, what do you think is going to happen when I put people on their back? The literature predicts that maybe it'll be a little bit tougher, but they should be able to do the task. And what we find is something that at least I found super surprising, which is that not only are they bad, they are almost 2 times as bad. And not only are they 2 times as bad, collectively as a group, there's no statistical change from beginning to end. For 40 minutes, they're stuck in this struggle where they cannot improve no matter how hard they try. It is a purgatory in many ways. To make you emotionally understand how crazy this is, 90% of people in this will say, I don't have an accurate sense of where I am in space. So this is a very surreal, weird environment to be immersed inside. Now to get some more insight, what I want to do is show you some raw data, because raw data has some secrets in it. And this is raw data for someone in upright, where they have a good sense of where they are. This is trial 20 at the end of the experiment. On the x-axis is just time while they're doing this experiment. And the green line is just the balance point, and all I want you to just quickly look at is what the black line is doing. It's just oscillating really closely around this green line. She's doing a great job. She has mastered this task. And maybe by now some of you are thinking, this guy promised me somewhere where I could collaborate, some connection to the work that Katya's doing. I don't see any of that right now. But wait, look at this graph. Look at it. there's something there that's similar to what Katya has shown. And that key word is oscillations. Oscillations. I have oscillations in position. I have oscillations in velocity. I have oscillations in joystick. Are there any generalizable patterns that exist within the oscillations I see in my experiment and what Katya sees in hers and what you see in yours? Are there any common set of tools that we can use together to characterize a type of oscillations? Is there a simple system that we could design that could reproduce the complexity that we see in our data? It is worth asking, because many of us, almost all of us, are working on oscillations. What kind of tools could we all share? Now, these are ideas and questions that a lot of people have already looked at. But again, if we create a common narrative that can collect all of the oscillations at Brandeis, how epic would that be? I, to study my oscillations, use something called the stabilogram diffusion function, which is an idea borrowed from physics of looking at a particle immersed in a field. Could that possibly be useful for the oscillations that you look at? I don't know. Worth investigating over a collaborative lunch event. Now let's get a little bit even crazier. If I take this trial data and I replot it in a position velocity phase plot, where on the x-axis we have position, on the y-axis we have velocity. And I'm not going to go into the detail of this because it's going to take a long time for me to explain. I do pull out a lot of mathematical stuff. But all I want you to be able to see from this is here's trial one. In trial one, they have huge position and velocity. They are making these huge undulations, these huge cycles, these huge oscillations. And on this plot, they are all oscillating around this green point. And by the time they get to trial 20, their oscillations become a lot tighter. But they're still centered around this green point. What could that green point be? It is the gravitational vertical. And another way of saying it is, what if that gravitational vertical is like, in a metaphorical way, an attractor? What if the gravitational vertical is an attractor and the system is oscillating around it? And if I use that vocabulary, maybe some physics people are now salivating. Maybe some math people are interested. Maybe some chemistry and biology people are interested. These vocabulary can allow us to explore maybe some common patterns that exist everywhere in all of our data. Now, I'm going to get even crazier. No one get angry. But maybe in a metaphorical way, this is like the idea of homeostasis. You have a homeostatic set point, which is a gravitational vertical, and people are trying to balance to it. The system causes you, in a way, to deviate, and then you try to pull yourself back to it. Maybe there's some shared ideas that we can do between the homeostatic community and the balance community. And now, what would happen if I took that attractor point? If I took that homeostatic set point and I broke it? That's interesting. That's a biological thing that could possibly happen. What happens if I break that attractor, that homeostatic set point? And now some of you might be saying, how is he ever going to break the gravitational vertical in his experiment? How is he going to get rid of that? And one idea, or I already explained it to you, is that in the back condition, you don't have that set point. When you're in the upright, you have a good sense of where that reference point is. But the moment I put you on your back, you don't have that reference point anymore. So what happens? Anything interesting? Anything relevant to a biological system? One thing that you know, and again, this is one of the key words that Katya was talking about, is that when people are in the back condition, I already showed you this graph before, the variability is a lot higher. But now here's a philosophical question for all of you. Is variability just noise?
Similar.
What's that?
The variabilities look very similar. Also, it's weird that the variability is small at first and then large at the
Yeah,
end.
that's actually interesting. So, and maybe we can talk more about that at the end. The variability is the error bars, but statistically these are two different groups.
Yeah, but they look the same with me.
But then the variability is much higher here. So what you're asking is there anything interesting going on with
No,
the
I'm saying that the
variability?
variabilities look similar between the width of the red is similar to the width of the red.
Yeah, but in order to characterize variability, the width is only telling you what is the range of the variability. But what is the flavor of the variability? So let me just show you. Let me just explain to you. Maybe this will satisfy what you're asking, because you're asking for something deeper, right? So what I'm going to show you next is this variability or noise just trash, or is there a structure? within that variability? Is there some structure in this variability that is different than the variability over here? My answer is yes. And what that difference is the following. This is raw data. This is people on the back. This is trial 20 at the end of the experiment. On the x-axis, if you remember, is this time. This black line is the angular position. The green line is the balance point. At first, she starts close to the balance point. But then, as time goes by, she begins to deviate. Her variability is high, but there's a structure inside it. And that structure is a structure of positional drifting. All subjects in my experiment that are on the back show a characteristic pattern of positional drifting. It's not just variability. It's not just the variability is roughly the same size. It is that these people are positionally drifting. And that's a fascinating finding, to me at least. I was not expecting that. I was just expecting the variability to go all over the place because they don't know where they are. So, just in the phase space, just to show interest here, here's upright. You can again see when you have that attractor state, it's all focused. But the moment you shatter it, you start tumbling down this position axis. Now, the positional drifting that we see, what's interesting about it is it suggests that the velocity signal that is going to the neural integrator is not being properly or adequately integrated for a dynamic stabilization task. And now here's something interesting that I bumped into in a review paper. And what these people say is that perhaps angular path integration occurs through a neural ring attractor system. Now this is not a subject that I've mastered. It's just something that I've bumped into. But what they say is that somewhere in the brain, you've got a bunch of attractors, and as you get more sensory information, you go from one attractor to another, and maybe that is how we integrate. And this is what they say, you don't have a strong calibratory sensory signal, you then have some sort of random drift in the system. Now, if that's really true, could that be the reason why my people are showing drifting? I don't know. But it's worth having a collaboratory lunch conversation on. And what will we do? Will we propose an experiment where we put a rat into the Hulk and stick an electrode in its brain? And if we do, where would we stick it in? And what would that experiment look like? I think it's fascinating. I think it's a place where I and you can connect together. So one mega cool idea is to create a idea generation series. And I don't say lecture, because I feel like lecture series makes us into knowledge retainers, but I want to be more. I want to be an idea generator. So one idea is to have this idea generation series where we find commonalities that span the research. And I've spoken to Bolin, who is a postdoc in the Miller lab, and he looks at two neurons or two groups with the most simplest connections of excitatory and inhibitory. And his question is, can he reproduce the complexities that we see in oscillatory and dynamical systems using this simple system? And from there, we can go and add the complexity and see if there is any larger pattern there. So if you're interested, contact me, because we'll probably totally do it. Now, I'm running out of time, so long story short, when I put people on the back, some people show individual differences, some people show a little bit of learning. And the postdoc fellowship that I wrote is application-based. Its purpose is to solve some of the problems that NASA has. And so one of the things I proposed was to be able to predict performance and learning at the end, looking at the beginning. And the way I'm doing it is I'm starting a collaboration with Peng-Yu Hong, who's a professor of computer science at Brandeis. Now the reason I bring this up, to make you guys salivate, is that as I was talking to Peng-Yu, we found a common dream between him and me, which was to create an idea generation series that brings together machine learning and science. So as some of you may know, I've done 4 series on topology and neuroscience, just organizing it, bringing those 2 departments together. And one of the cool things that came out of it is Sarah and Daniel began thinking about how to use machine learning to understand patterns of branching within her data, of her neurons. Now what blew my mind is when I went to Peng-Yu and started discussing with him, he's already actually started doing this with a collaborator outside of Brandeis. And I'm like, why? We're right here. Why are we not collaborating? If you guys want to work on this, create a relationship with the machine learning community, contact me, because we're going to try to do that. And the last quick idea that I want to share with you is I propose creating a brain-machine center at Brandeis for a provost grant, which got rejected. Now, the problem with identifying Brandeis is that we don't have an engineering school here. And to me, at least, that limits a lot of different things. it limits the way for us to create translational and application-based research and for startups and stuff like that. So my proposal is that we create partnerships between the Maker Lab, the newly forming Automation Lab, Computer Sciences and Sciences, and a variety of sensors. So if you're interested in that, contact me. And I'm doing stuff with high school kids, so if you're interested in that, you can contact me as well. Okay, there are other storylines, and I'm almost out of time, but if you're interested in just, exploring these ideas, you can ask me. One is creating a training program to allow people to dynamically stabilize when they're on their back. And the other is to understand why is it that these individual differences work. And the notion there, and I haven't actually started working on this part yet, is to look at the most basic thing. Look at their vestibular threshold. Does that correlate with performance? Then go to the next level. What about looking at the internal model that allows them to perceive where the balance point is? Maybe that's it. Or maybe it is skilled motor learning. So that's going to be the battery of tests that I will use to assess the individual differences in these people. This is my lab. Thank you to all of them. And any questions?